Okay, we'll make a start then. So today we're going to talk about the new microbiology program that we are launching. Basically, the first sample is due this week, and we'll be running it probably tomorrow. So so far, the pilot study that we had went well. So we had about 130, 140 participants that actually participated in the pilot study. So hopefully we'll get the majority of them on over the next few weeks and months. Understand that maybe at the moment it's not the ideal time to start a new program, but there we go. This is it. So look, try our best to introduce this to you. I know this will be new to a lot of you. It's new to us as well. So hopefully together we'll work our way through it. And this will make a very useful additional program to the portfolio that we have. So I said last week this presentation would take about 20 to 30 minutes, and we finished up taking an hour with questions at the end. So hopefully we won't take as long as that today, but we'll move through it. If you do have any questions, please take a note of them, and we'll go through them at the end. You can send them through into the comments section, or else if you just want to discuss it at the end, that's fine. So let's move on. So for an overview of the actual program itself, the kits will contain 3 samples, the 2 distributions per year. So basically running 1 sample every 2 months. The case studies that are required are on the RIQAS Net kit insert section. So the case studies that the labs will need to basically help them to know what's the correct medium for them to grow up the strain, you'll need to refer to RIQAS Net and for the kit insert section, they're already there. So the samples themselves are generally samples or strains from the National Collection of Type Cultures, which is a UK-based body. There are other possible strains, say TCC and CDC or 2, just to name. Now, we may use some of those strains moving forward in the future, but initially anyway, we'll be using NCTC strains. The way that the strains are reported can be basically looked at 3 different levels. So we'll be looking to see whether they're gram positive or negative. We want the genus and then the species. Not all labs will do it to species level, but that's fine as long as that's their protocol, and they then record that that's the case actually when they're submitting their results. We do offer the basically multiple registrations the same way as we would with our normal programs. This can be useful for training purposes, basically for helping new members of staff to assess their ability to correctly identify the organisms. Now, we understand that nowadays many labs will just use automated systems, but I do believe that in many cases they still have to be able to basically to identify going through the old traditional route. So there is a potential there as well for that then. So you can register multiple times. So once for the traditional route, looking at under microscope, for example, or otherwise then using a more automated system. Okay, so you do have multiple methods now. We were asked last week what's the maximum number. Initially, we are starting with 5, which is our just normal for the program. But if you do have a large organization that requires many more registrations, then we can look into that separately. So 5 initially, but so we can look outside of that as well. Okay, so onto the samples themselves. The samples will come in these foil pouches. Now, it's only the pouch itself which is labeled. The sample, this tube, which is contained within the pouch, is not labeled. So once you take the sample out of the foil pouch, it's not identified. So it's important that the sample number is correctly recorded actually on the agar plate whenever basically that is streaked out. So it comes basically in 2 parts. There's the actual pellet, which is a lyophilized pellet for the microorganism at the bottom of the tube. At the top of the tube, there is a pre-filled ampule of hydrating fluid, so which is then used for the reconstitution. I'm going to take you through here now a visual presentation of how the samples are prepared. So initially we go from obviously, as I say, opening the foil pouch, but before we do that, we let the sample equilibrate to room temperature. So obviously taking it out of the fridge, let it equilibrate. Then we remove the device. Now, as I said before, it's only the actual pouch itself which is labeled. So please remember that to record the sample number on the primary culture plate before before the sample is opened. Should I say, don't disassemble the device until the actual sample is hydrated, because obviously you'll see in the next step, once you crack the ampule in the top of the device, obviously the hydrating fluid then leaks down into and hydrates the lyophilized pellet at the bottom of the tube. So it's important that the sample is not opened until that is done. So next step is to crack the ampule. So as I say, that's at the top of the tube. So basically, it's very easy. So just pinch it between your fingers. That will crack the ampule, and then the liquid itself then will fall down and hydrate the pellet. Just to make sure you get any drops, we do recommend that you tap it on a hard surface, just so that any droplets which are maybe clinging to the side of the tubes, that they will fall down and then hydrate the actual sample itself. At that stage, basically use a pinching motion to make sure that the actual pellet is really broken up and the suspension then is homogeneous. So really want to get obviously the lyophilized pellet hydrated and make sure that it forms a nice suspension. Now, at this stage, we don't have any recommendation whether that should be 30 seconds or minutes. We don't have that recommendation at this stage, but just for a sufficient amount of time that whenever you're pinching it between your thumb and finger, that you can feel that the pellet is completely sort of broken down. Step 6 is then basically to open the tube. So the tube's in 2 parts. The top part that had the ampule then also contains then the swab. So the swab at the bottom then will be soaking in the hydrated, basically where the pellet was, so not a hydrated sample. So you want to make sure that that heavily saturated swab is then used, well, basically straight away, so that it doesn't, so that any of the hydrating fluid doesn't evaporate off. So really want to, once it's reconstituted, basically take and homogeneous, then take it out and use that to inoculate the actual plate itself. So normally what it's recommended then is you inoculate the basically a third of the plate, and then you use a sterile loop to streak out to facilitate then colony isolation. Remember that this is a biohazardous material, so they are, even though they come in a lyophilized form, it is potentially, well, will cause disease. Potential, it has that potential anyway. So these should not be opened outside of a lab that has the ability to deal with biohazardous material. Now, most of the samples that we're providing will be biohazard level 1 or 2. The list that we provide of potential microorganisms, there are some in there with higher biohazard levels. We won't be providing them, well, not initially, certainly. The early stages of the program will be providing just biohazard levels 1 and 2 samples. So once you get to the stage of basically plating out the microorganism, then you should incubate that. And normally it's incubated inverted, and that's then used at the correct temperature and conditions. How do you know what those correct temperature and conditions are? That's where the kit insert comes from, because basically this will sort of give the labs the information that they need to know what sort of temperature and conditions that would be suitable for a microorganism that is found in those conditions. Okay, so that's then basically getting the sample, plating it out, and so what you're going to do now is to, well, obviously report the results back to us, which is done via RIQAS as normal. The reports that then will be sent back is what we're going to spend the next 10-15 minutes looking at. So some of these screen shots that we've taken, some of the text might be quite small to look at on screen. I do have zoomed in sections, but they may still be quite difficult for you to read. We do have a report example, so we can send that to you. Remember as well that there is already provided by marketing. There's a flyer already provided, basically taking through this information. So that is also available. So this is really just for us to go through in a bit more detail. And if you have any questions, then it's time to basically ask those questions. So first page is broken down into 6 sections, and this is all regarding the current sample as opposed to historical information, which is going to come on later on. Should I mention as well at this stage that the reports are broken down into 2 parts. First of all, you'll have the bacterial identification, and then you'll have the antimicrobial susceptibility testing aspect. Now, some other EQA providers will provide these as 2 separate programs. So you'll have a bacterial identification program, and then you'll have the AST, the antimicrobial susceptibility testing program. We are providing them as one package, just some a bit more benefit for the actual customers who are participating. So first section is the bacterial identification for basically looking just at the current stats. So very top is we have a line basically saying the number of expected organisms. Now, once again, we're starting off slow with this scheme where we're only going to be providing a single organism per sample. Moving forward in the future, there is a potential that we may offer multiple samples, or should I say multiple organisms per sample, or an organism with a background flora. But initially, we're only going to be providing a single organism, and this is what's recorded at the very top of the page. Then we have the expected organism. Obviously, we know which we're providing. So in this case, it's a known organism that we're providing. So in this case, the Pseudomonas aeruginosa is being provided. This is one that we had in the pilot study that was sent out. So we have the expected organism and the result from the actual customer itself. Here we see as well whether the result was sent for a referral. Now, what does that mean? Well, basically, some labs are not kitted out to deal with a certain strains, certain genus and species. So what they'll do is once they think it's heading down a certain line that or certain organism that they're not set up to deal with, i.e., an organism probably with a higher biohazard level. Then what they would do is to send that out for a referral to another lab. If this is part of their protocol, then that's fine. They'll still then, what we'll see here in the next section, they'll still get scored based on the results which are coming back from that referral lab, but they'll be scored at a lower value because they didn't actually do that themselves. So obviously, moving down the line, it would be useful if we actually had the referral lab on the scheme as well, and you had both. But it's useful information for obviously the lab which is sending it out to referral, because obviously they're wanting to see is the information that referral lab providing, is that correct? So at least once again, the lab will see how the referral lab is actually performing on their behalf. Next section then is where we keep the case study. So that's just recorded in the next section down. Quickly followed then by the actual overall scores. So on the left-hand side, we see the score for this sample. Now, the scoring section's on the next slide here, but just to say at this stage, so it's scored out of 3 if the lab identifies it themselves. Here we then see the average scores based on the global participants and also by country. And that's broken down by the method that was in use, and then just overall, basically all methods. So that's the next section. The actual scores is basically indicated here. So as I mentioned before, there are 3 levels. So they can do it by the gram stain, which is a positive negative value. They'll have then the correct genus and then the correct species. If they get all 3 correct, then it's a score of 3. That is if they don't send the sample for a referral, i.e. they make the identification themselves, which is fine. It's also fine if they leave the species blank. If that is their protocol, so that if the case is that they only ever identify to the genus, then that's fine. They'll get still full scores for that, but they have to record that as their protocol. If it's not their protocol and they leave it blank, or that they get the incorrect species, then their value or their score drops down to 1. So if they get the correct genus, then they'll get a score of 1. If the genus and species is wrong, then it's a score of 0. That's if they get the correct gram stain. If they get everything wrong, then it's a score of -1. Now, once again, that's just to highlight that is if they don't send the sample out for a referral. If they do send it out for a referral, then the score is slightly different. It's only marked out of 2. Most of the scores are the same, as I say, apart from the top scores, which is, say, that lower score of 2. But then an incorrect score is actually 0. Because it wasn't them who actually got it wrong, then they get a score of 0, so they're not marked negatively. Okay, so that's the way the scoring works out. Next section down, for section 4 out of the 6 is basically a breakdown of all the organisms that were actually reported to us for that sample. So here you can see the number of participants that got correct results. The black triangle basically denotes the participants' response, and it's in order of descending frequency. So once again, the expected organism then is recorded just above that chart to say, once again, just to highlight what the correct organism was. Next section down is where we're showing basically the breakdown by method, and then the number of correct partial scores or incorrect size scores. It's the partial evaluations or incorrect evaluations for this individual sample. Okay, once again, triangle denotes the participant's response, and once again, it's sorted in descending frequency. So initially, this is basically based on the information that we had from the pilot study that we ran. So we won't have a full list of methods, and as you can see as well, we've introduced some like the Vitek 2, all the models, the Becton Dickinson Phoenix. We have all the models grouped together. As the program expands and we get more and more participants, then we will break that down and provide more further information into some of the various instrument systems. As you can see there as well, we don't have any of the Molotov methods, but that is because we didn't have any actually for the actual pilot study, but that's certainly something that we can add on. So basically, if there's a method or an instrument which the lab is using, which we currently don't have, just please let us know and we can look at adding them on, the same as what we would do for the normal programs. Okay, and the last section on the first page is really the same information again, but it's broken down into numerical and into percentages. So as you can see, the percentages are going across the row, and it's basically for each of the individual methods. So here we can see for the conventional tests, we had 81 correct responses, making 90%, 6.7% for partial, and 3.3% for incorrect evaluations. Just as a reminder, partial, what does partial mean? It just means that they haven't provided the full genus and species. It's only been the actual genus that they have provided. Okay. A correct is where they've provided the genus, species, and also then genus if it's their protocol to do that. Okay, so that's the first page. The next section will be over the page. It's the historical information, and this is broken down into 4 sections. So we'll look at each section individually. So the first section is the historical performance just for the individual lab. And this takes the individual score that they've had for each sample. So it'll start, obviously, very first sample. The maximum score will be 3 out of 3. So that's the top score for one sample. So covering it over, obviously, over 6 samples, then once you achieve 6 samples, then the maximum score will be 18 out of 18. So that'll be your 100%. Once again, if you send samples out for a referral, then the maximum that you would have would be out of 12. So it would be 12 out of 12 would be a maximum score in that case. But you're still then looking at a percentage of 100%, for example. So that's your historical performance. Next section over is then where we're looking at the historical performance, basically then looking at globally and per country. And that's based on the method once again. So you can see the various percentages. Now you'll see the symbol here for the open circle and the triangle. This will be shown then in section 4 once we get to it. But section 3 is just really almost like a Levy-Jennings chart, but it's not. Um, but basically going, just showing how the lab has performed for each individual sample. Current sample, obviously on the right-hand side, just like we normally do with our normal reports, moving over in time. Unlike obviously any of their quantitative reports, there's no quantitative value here. So what we're displaying is the actual organism that was provided for that sample. So you can see for each individual sample, each individual organism, how did they score for that in that case. Last section is really where, as I say, where we're displaying that information, the lab's performance compared to the historic performance for global all methods and global method. We're not displaying the country scores on this chart, so it's only the participant against the global all methods and the global method, or the method that the participant's actually using. Obviously, then the three icons, or the three symbols, are what you get displayed on the actual chart. Now, just one thing to mention on this chart here is really whenever you're looking at each individual sample. So this will change over time. So obviously we're looking at changes of performance overall. So if there is an organism which is particularly difficult for the labs to score, then it has the potential then of obviously changing this percentage. So obviously the percentage will drop. Then obviously moving on to the next sample, because it's an average and it's a rolling window, then it'll take time for those scores to build back up again. So please remember, so this is not the score just for this individual sample. This is the score for an average out of the rolling window. Okay, so just to make sure that's highlighted there. So it's a rolling average score. It's not the score just for that individual sample. Okay. Oh, sorry, I've gone the opposite direction. So that's the end of the identification part, which is really just the first 2 pages of your report. Then we move on to the second section, which is the antimicrobial susceptibility testing. Just to highlight what that is for those who don't know, Kirby and Bauer first brought this sort of method up. I can't remember what year it was, but this was for a long time sort of the standard method then for antimicrobial susceptibility testing. Basically, you take they're basically working from left to right here. So you take the inoculated agar plate, which has your microbe on it. You place on that different discs of antibiotics at a standard concentration. Then you incubate that overnight, and what you'll see then are these clear areas appearing around the actual discs, so these zone of inhibition. So you'll have basically as highlighted here, you'll have the disc, you'll have basically an area where there's no growth of microbe because the microbe is susceptible to the antibiotic. And then basically around the outside, you'll have the bacteria, which is still growing. So what you do then is you measure the size of this area and record that basically, and this will give you what if the microbe is susceptible to that antibiotic or not. But it's not very, should I say, it's useful to see what antibiotic is useful against a certain microbe, but it doesn't give you basically what we're looking at here in the next slide. It doesn't give you this MIC, the minimum inhibitory concentration, which is the lowest concentration required. Obviously, in today's age where you're trying to restrict the use of antibiotics, you want to basically, and some antibiotics are actually quite toxic to patients at a certain concentration. So you want to minimize the amount of antibiotics that you're actually treating a patient with. So what we're looking at here is this minimum inhibitory concentration, or this MIC. And what we then do is use that basically along the lines that with what we've had before to classify whether a microbe is susceptible, whether it's intermediate, susceptible, or it's resistant based on these MIC breakpoints. So really the breakpoint is the chosen concentration where an antibiotic, which defines whether a species of bacteria is susceptible or resistant to the antibiotic. And really what we're saying then is that if the MIC is less than or equal to this breakpoint, then the bacteria is considered susceptible to the antibiotic. Now, these MICs basically are categorized by various international agencies. So the two main ones are I've mentioned here, and they work out these breakpoints based on different methods and different mechanisms. So the CLSI, which is an American one, basically provides the most popular guidelines. These are based on pharmacokinetic, pharmacodynamic, this PK/PD, based on those properties and then mechanisms of resistance. You have then the European body, this UCAST, which also uses the PK/PD properties, but also then uses these epidemiological MIC cutoffs or ECOFs. These are determined by, as I say, this European committee. Now, these cutoffs and these breakpoints do get updated generally on an annual basis. So it's important for us to know which guidelines that the customers are using and if they're using what version that they're using. Initially, whenever we first run the pilot, we just had it broken down by CLSI or UCAST. There are other country-specific guidelines as well, but the two main ones in use were the CLSI and UCAST. But as I say, they do have different versions, and it was clear then after we ran the pilot that we need to break down the guidelines into the various versions. So we will be introducing that then moving forward because they do get updated. One of the antibiotics that we provided, one of the antibiotics against the organism which we provided in the pilot then actually had changed and there were new recommendations. And it was clear that some labs were using the old recommendations, some labs were using the new recommendations because we had labs that were reporting this I for the intermediate level, which apparently now doesn't exist. Sorry, I'm going to get this wrong. It wasn't that the I didn't exist. It was that none should be down for susceptible. It should only be resistant or I. You shouldn't have any susceptible results for this particular antibiotic, which is what we had. So it's important, as I say, that we're using the guideline, and the guidelines are what we then are using in this next section. So why I put those couple of slides up was just really to highlight the importance of these MIC and the guidelines that then the labs use to determine the susceptibility of the organism to the antibiotic. They will be different. As I said, their last point there, the breakpoints then are recommended, the recommended by UCAST are generally higher than the CLSI. So you will get potentially different results depending on what guidelines then are in use in the lab. So how that gets all presented then on the report is basically like this, or as you say on the left-hand side. So what we have here is a list of all the antibiotics that were reported for this individual organism. So for Pseudomonas aeruginosa, basically we had this long list. It was actually goes on to another page of antibiotics. Now, not all labs will use all these antibiotics. That's not the case at all. Um, it will be that they will have, as part of their lab protocol, a set number of antibiotics that they will test for. But it's useful information for the labs to see then what other labs that are using the same guidelines as themselves, what antibiotics they're using in their situation, in the clinical situation, for treatment. So it obviously provide, and they can see really as well in how useful that they are, whether they are provide outside the antibiotic is susceptible or not. Sorry, not the antibiotic, but whether the microbe. Let's call that, whether the microbe is susceptible to the antibiotic or not. Sorry about that. Okay. So basically, for each antibiotic, which is listed down the left-hand side, you'll have it broken down by what the labs have submitted. So whether they say they're resistant, it's intermediate, or whether it's sensitive to the antibiotic. Now, as you can see, we're using different terms here. Some of the terms have and are changing. So instead of calling them sensitive, some labs will call it susceptible. And in terms of intermediate, that will now be intermediate or susceptible. So there'll be a mixture of terminology being used. Not all labs have changed over as yet. This is the older terminology, but a lot of labs have now moved over to, instead of calling it sensitive, it'll be susceptible. So we have basically what the labs have reported. And then we have, for this individual lab, we have what their results were. So whether they have said that it's sensitive or intermediate or resistant to the antibiotic. So you don't have that obviously for each antibiotic, because this participant actually doesn't use all those antibiotics. So it's only what you have listed here are the antibiotics that they are currently using or test for in their lab. We'll come to the score just in a second, because that is based on then the target. So how do we determine what the target is? Well, what we're using then is a consensus, same way as we do for most of our other programs. And in this case here, the target is based on an 80% agreement, or at least 30% more than the next common response. We do then require at least 5 responses as well, or else this basically what we record here is that there's too few. Okay. So we do have then this in brackets, which is a Y and an A. So if the target is based initially on the participant guideline, you'll have a Y for your guideline, followed basically by all guidelines. So if there is not an agreement, then for the participant's own guideline, we will move up to then the all guidelines. So here we see against the target whether this Y, for example, so this target of sensitive is based on their guideline, whereas we do have a few where there was insufficient information, so it's gone up to the all guidelines. Now, it is, as I mentioned before, because the guidelines do differ, then you will potentially get some different information, and that's important to look out for. We may restrict that in future. As we say, this is a new program for us. We're learning this ourselves, basically as we're progressing with it, and we're getting feedback from the participants as the scheme progresses. So some of the feedback that we've had so far is whether we should move up to the all guidelines for some antibiotics, because as I mentioned before, some of the guidelines are quite clear where maybe you shouldn't have a susceptible result at all. And in that case, then maybe it's not relative to go up to then on all guidelines. So we'll gradually work our way through those as time progresses and we get feedback from the participants and also obviously more information ourselves. But initially, that is the way the process is working. But if it does go up to the A, we just need to be careful that some of the guidelines may not match. So that's the way it basically works out. So the actual scoring then is, you'll see that basically on this next slide. So if the target that we have assigned is sensitive, and then the participant says it's sensitive, then you'll get a score of 2. So it's 2 out of 2. If they say it's intermediate or intermediate susceptible, it's a score of 1. If they're saying it's resistant, whereas it is sensitive or susceptible, then they get a score of 0. Slightly different than if it's a resistant organism. So if we're providing an organism which is resistant to that antibiotic, then if they're saying it's susceptible, then they'll get a score of -1. If they agree and it's resistant, they'll get a score of 2 because it's correct. And if it's intermediate, then it's a score of 1. Slightly different again for an intermediate target, where you've got an intermediate is 2, and you'll have a score of 1 for each for the 2 other reported results. If there's no scoring possible, then that is, if there's no scoring possible if there's too few or if it's NA. So that's the way the scoring works for the AST. And this is that covers over basically 2 pages. At the bottom of the next section then is a scoring summary where basically we just total that all up. So all the scores for the individual participant. So if they've reported 10 antibiotics, then they'll be out of 20 is the maximum score. And then what they have actually achieved, so it'll be 19 out of 20 with a percentage, 95%. Then we break that down for the actual guideline. So once we look at all the antibiotics that have been reported for the similar guidelines, and then once again, it's very similar for all guidelines as well. We do give a small summary then of just basically stating that 3 of their antibiotics have no target and are not scored, if that was the case. So there's a summary then for basically all the antibiotics that get reported for that individual participant. The next section and the last section that we provide in the susceptibility testing is really where we're just breaking that information down. There's no scoring in this. It's just really to help to see basically the differences in the guidelines. As I said before, especially if we're looking at the all guidelines, there may be differences. Where you would then go to is the section. So for this antibiotic, you're able to look at the individual guideline and you can see then the responses. So here you can see there's quite a significant difference between the CLSI and the UCAS guidelines, where the CLSI basically are saying that this is sensitive, that this antibiotic, or sorry, the microbe is sensitive to the antibiotic, whereas the UCAS guidelines are saying that it's actually an intermediate. Although we do have still a few labs that are saying it's sensitive as well. The unspecified, we believe, will probably, once the actual program develops, there'll be less and less people that are unspecified. We had to record that in the pilot like that, just basically because labs actually, as I say, didn't specify their guidelines. But that is the important aspect of this antimicrobial susceptibility testing. Now, when some EQA providers will actually provide the actual mic points, they'll actually provide sort of the actual, basically a breakdown of the gap that gets provided for each antibiotic. We haven't gone down that line, not at this stage. It is obviously maybe a possibility for the future, but at this stage, we don't think that that's as relevant as this information here that we're currently providing.